Okay, very good. I'm almost there. Let me get my clicker. Thank you for your patience. Sorbent. I'm so excited to learn about sorbent. Okay, let's do this. Yeah, all right. Okay, so what's the main principle used in immunoassay? Very simple. Antigen react with, that's it. Very good. So it's based on antigen reacting with antibody. Application? Yeah, protein for what? Allergens is one. What else? Adulteration, like meat adulteration, when you have different types of species of meat that they shouldn't be there. What else? GMOs. Yes, GMOs, absolutely. So all of this is protein related. Allergens, adulteration or authentication of meat, GMO, but also we can use ELISA to, or immunoassay, to detect viruses and bacteria. Also, they can be used for small molecules like pesticides, antibiotics, the toxins like aflatoxin. So, what is an epitope? Where the antibody binds to the outside of the antigen. Very good. So it's a region on the antigen that the antibody recognizes and binds to. Very good. Okay. Do you remember polyclonal, monoclonal antibody? So our body, when we are, when there is a foreign body that comes in, and if we are told to be attracted to this foreign body, the B cells, we have so many different types of B cell, will produce a whole lot of antibodies that will recognize different epitopes. So they don't recognize the same epitope. They can be attracted to this antigen at multiple epitopes. So we call them polyclonal. Monoclonal are lab produced. So they that one kind of B cell is proliferated in a way that it will secrete only one type of antibody that will recognize the same epitope, the same region. And they're very, very useful for kits. When we buy ELISA kits, most of the time, or all of the time, the antibodies are monoclonal. They recognize one type of epitope. Somebody was talking. Outside. Oh, should I use the microphone? No, you're all good without it? Okay. And then, okay. Do you remember difference between direct and indirect ELISA? It was complicated, Emma. Wonderful. Very good. Thank you. So this is the image, and I want you to remember that. So we have the primary antibody would be the one that would recognize the antigen. If it is labeled with the enzyme, labeled meaning the enzyme is bound to the antibody. If it is labeled with the enzyme that we use to add a substrate and produce a product that we can determine absorption, then that's direct action. So your antibody is labeled. When the capture antibody or the primary antibody is not labeled, but we buy an antibody that is labeled, but this antibody recognizes this unlabeled antibody. So the primary antibody becomes the antigen to the labeled antibody. Now, I ended the lecture there last time, and I told you why we do that, because it's easier to buy an already labeled antibody rather than you label it in the lab yourself. So this would be an available reagent, and we buy one that would recognize the primary antibody that we are using. So let's say, give you an example. I'm allergic to peanut, which I'm not, but in case you're interested. So let's say I'm allergic to peanut. I'm determining if I change peanut protein in a way that will no longer be allergenic to me. Like I hydrolyze the peanut protein completely, destroy epitopes, and now I want to determine if I'm still allergic to it. So a sample of the blood, the serum of that sample will have antibodies. I'll take those antibodies and then I'll see if they will react with the peanut protein now that I hydrolyzed. But it's so hard to take human serum antibody and label it. It will take forever to get that, chemical reaction happen. So I go and buy a reagent that has an antibody for human antibody. So the human antibody becomes the allergen, the big antigen. Okay. How do I produce an antibody to human antibody? It would be, okay, I'll take a goat. Somebody will take a goat and will inject in the bloodstream of the goat antibodies from human. They will recognize it as foreign and they will produce antibodies to it. Collect serum and then take those antibody, purify them and label them, and they become the agents for sale. Okay. Yes. No, we'll talk about that. Sometimes you, like in sandwich ELISA, you have the capture antibody that will capture the antigen, and then that's sandwiched by the primary or detection antibody. We'll talk about that. Okay. So main steps, I'm going to draw this so that you can visualize it with, maybe I'll use this other sword. Okay. So we always run ELISA in a plate. So consider this is a well, one well. Okay. So one tiny well of 96 wells. So the plastic has a hydrophobic surface. So let's say we have a sandwich ELISA. We will put first our capture antibodies. So remember, those antibodies are proteins. So they will bind the hydrophobic interaction to the surface. And then we do a wash step. Always in ELISA, put the reagent and then you wash. Everything not bound will be washed out. So now we have our antibody, we call it capture antibody. And we wash. Then we do a block. What does that mean? We use usually albumin, a serum albumin, bovine serum. BSA, bovine serum albumin, a protein. We want all of the surface to be covered by non-reactive protein so that we don't have non-specific interaction with the antibody. So we block, call it block. with a non-specific protein. Then we wash. After every step, there is wash. So anything not bound, we wash it off. So usually plate feeders have a washer specific for this kind of method. So we put the reagent, we wash, and then now we put the entity or the sample that has the entity. So they will bind to the antibody. And then we do a wash, any excess will be washed off. And then we put our primary antibody or detection antibody. So doing direct ELISA with direct detection antibody. So it will be antibody that is labeled. Let's just do the direct for that. So it's labeled with the enzyme. And then you do a wash. Every time with anything, let it react and then wash. Then you put substrate. There's no room to go up. So wash, then substrate, and once we wash again, everything non-reactive will go away, and then we let it incubate so that the substrate converts to product and we measure absorption. That's it. These are the primary steps. Question, yes, Alan? The blocking is very important because if you don't block, you will have a non-specific interaction rather than interacting with your antigen or the interacting with your capture antibody. They would interact with the surface. It's not specific. So you don't want that to happen. So we don't do that. That's why we have a block. All right. Any other questions about those steps? Very good. So this is just to capture exactly what I had here. So this is your direct sandwich ELISA, where I might ask you to label. I might give you in an exam an image and say label. So this would be your capture antibody. This is the antigen. This is the primary labeled antibody. Okay, so here in A, we have capture antibody, antigen, detection or primary antibody that is labeled with the enzyme. Sometimes to enhance the sensitivity, we get antibodies that are biotinylated and have streptavidin conjugated with enzyme. You don't have to remember those names, but what that means, you have an antibody that has multiple sites with a lot of enzymes. This will enhance the sensitivity because then you have a lot of enzyme ready and bound to only one end. So you are, making sure that you can detect those antigens very low concentration because now you have a lot of enzymes. So the substrate will have a lot of products generated. So this is just to enhance the sensitivity. So these are the purpose of it. You don't have to remember the names, but this is just to show that you have a lot of enzymes that could be bound to one antibody. In this C, this is an indirect sandwich ELISA, where this is the primary and this is the secondary labeled antibody. So primary antibody and secondary labeled antibody also with a lot of enzymes coupled. So a lot of sensitivity here. So very high sensitivity. And again, in any immunoreactivity assay, like we studied allergenicity in soybean, we always purchase this kind of labeled antibody that has a lot of enzymes integrated. It enhances sensitivity of the reaction. So this C is very, very common because of the availability in the market of such a labeled antibody. So that is very, very common. If you ever have to do immunoassays, you will go that route. All right. So this is an image, another image of a sandwich ELISA. So it just depicts, your solid phase, which is, this is your plate, your capture antibody, and then you put a sample that has the antigen. This red shape here is your antigen, and then when you do wash, everything in the samples get washed off. Then you put your labeled antibody and then your substrate and color generation. And this is just showing you how you would label such an image. Okay. This is another image to kind of emphasize the indirect sandwich ELISA, where you buy an antibody that is labeled that would recognize the primary antibody as an N. So it's just showing you another image of that with the enzyme and the substrate and all. Now I'm going to go into competitive ELISA. So far, we talked about non-competitive ELISA. Competitive ELISA is specific for small molecules. If in the exam, I give you an image for a competitive ELISA and ask you to give me an example of an antigen, if you say protein, it's wrong. This is specific for small molecules like aflatoxin, antibiotics, pesticides. So any small molecule that is of a small molecular weight can be recognized with this competitive ELISA. So what this is saying is if the molecule is small, it cannot be recognized on its own as an antigen. You need a carrier. So usually you have a carrier that is non-reactive protein, and together the carrier and the antigen is called a hapten. So small molecules require this carrier, and the carrier is not your target, just carrying your molecule of interest. So hapten is generally a general protein carrying or attached to your small molecule. So let me show you now how it would look like. So you have two options. Either the hapten is the first thing you put in the well and it binds the protein, the carrier protein binds to the hydrophobic surface, or you put your capture antibody first. So bound antibody, bound hapten. So what you see here is you see this whole thing is your hapten carrier protein with your target that is labeled. So this does not come from your sample. This is a reagent. This triangle here, free, comes from your sample. So what happens is your free molecule that you're trying to detect in your sample will compete with the molecule that is attached to this labeled hapten. So that's why it's called competitive. So this here will compete with the free molecule. The free molecule, all of it is an epitope. The whole molecule is so small that the whole molecule is an epitope. So it would bind, if it's present in your sample, it would bind. If it's not present, your reagent that is labeled will bind instead. That's why it's called competitive. So your reagent that has the hapten that is labeled with the enzyme will compete with any molecule you have in your sample. If you have high concentration of those molecules in your sample, less of those labeled ones will bind. So what that means is if you have high concentration, let's say, of aflatoxin, less labeled hapten will bind, so you will have less absorbance. So you have an inverse relationship between the level or extent of absorbance and the concentration in your sample. So that's why it's called comparative. They're competing. If you don't have any aflatoxin in your sample, let's say, all of these labeled ones will bind and will have high absorbance. So this is different than your regular sandwich ELISA, where the high absorbance is low concentration because they're competing. Okay, so that's why the word competitive comes in. And here it's the same thing. Instead, you have the hapten that is bound, and then you have an antibody that is labeled. So when you put your sample in the antibody, if you have a lot of aflatoxin in the sample, they will bind to those labeled antibody and they will be washed off. If you don't have a lot of aflatoxin, let's say in your sample, then the labeled antibody will bind to this hapten and then you have high absorption. So again, it's competition. So same principle, but here you have capture antibody bound, and here you have the hapten, the carrier protein, with the molecule of interest bound. And that is the reagent that you get, whereas these three triangles are what the molecule you have in your sample. Any questions? Emma? Yeah, you have a question. Maybe your question will help clarify to other students. Tell me but raise your vote. Oh, I just want to know why it's so. Oh, because the whole molecule is one atom. So there is no room for another antibody to bind. Once they bind to one antibody, there is no room for another one to bind. So they cannot use inside the ones. But think about a protein, it's a large molecule and they have multiple epitopes. So if one region will bind and another antibody can come and bind for another region of that. Right? So for sandwich ELISA, if you got that question in an exam, then you can say an allergen, a GMO, authenticity of a protein, you can use that as an example. If you tell me aflatoxin for a sandwich ELISA, I'm going to count it wrong because aflatoxin cannot be sandwich. Doesn't have to happen. All right? Okay, good. And that's the aflatoxin detection example. So if you have no toxin, no aflatoxin, all of the labeled ones will bind and have really high absorbance. So no aflatoxin. If you have aflatoxin, then more of that free aflatoxin in the sample will bind to the antibody and less labeled ones will bind, so you'll have low absorption. So in comparative ELISA, high absorption means low molecule of interest. Low absorption is high, so it's the opposite. Okay. Are we good? All right. So immunoaffinity chromatography is usually common to purify antigen or purify antibody. So it's an affinity chromatography. If you remember from chromatography, that is a very specific interaction. So your stationary phase would be made up of specific antibody to a protein or enzyme or any molecule that you're trying to separate from a group in a sample mixture. So you would just put your sample and only the antigen will bind and everything else will leave the column. And then if we want to collect that antigen, we just change the conditions of the bottle, pH, ionic charge or ionic content, so that it will be less interacting with the antibody and then need to collect and cure antigen. Lateral flow strip, maybe later in life, you might encounter it in pregnancy tests. So, and another application is GMO on the field. So what happens here, the principle is sandwich ELISA. Absolutely sandwich ELISA, how it is formed. So your strip have two regions. The first region here that you dip is having an antibody with a color attached to it. And then here you have a fixed antibody that will sandwich the antigen. So what happens is when you dip it in your sample, if you have the antigen, for example, in pregnancy, there's specific protein. In GMO, there will be a GMO modified protein. Will interact with this colored or color labeled antibody and then moves up the strip. And if the antigen is present, they will bind to this other antibody that is fixed here, and then you will have a positive test. If you don't have the antigen, the antibody will continue to the control line, and there will be an antibody to the labeled antibody, and then you will see a color. In both cases, whether it's positive or negative test, the control line should light up for you to confirm that the strip is not blocked. But if you see this test line, that means positive. You have a GMO, for example, protein that got captured by this fixed antibody, and then you see a color. That's the principle. Yes, Emma. How the color will join. Because here in this strip, do you see that strip, blue strip? It has antibody that is labeled with a color. What is it? The antigen is in your sample. See this like diamond shape? So any sample should have that. May or may not have it. Like, so this color is yellow here, but it's not indicative of anything. But if it's a pregnancy, you would have a specific protein in the urine, and that would be reactive with this antibody, and then it will move up this. And this antibody that has this antigen will be captured by another antibody sandwich at that test line. And then you'll see the blue color, whatever. So is it two separate? No, there's one antigen, but two separate antibodies. Oh. It's a sandwich ELISA. Okay. So you have the captured antibody that is labeled with a color, and then it goes to another antibody that will just fix it, sandwiching. If you have the antigen, you have the GMO protein, it will bind, and then you'll see a test line. If you don't, you won't see a test line, but you will see a control, because no matter what, this at the control, there is an antibody for this label. All right? Okay. Last, I want to talk about a Western blot. So Western blot is a qualitative. So if you have a lot of samples and you just want to see if you have immunoreactivity with any proteins in these samples. So what you would do is you would run a 2D electrophoresis that we talked about in the protein characterization lecture. So you would separate the proteins and you would have a lot of dots of protein. You transfer them into a membrane that is specific and tolerant of the agent. So you would use a transfer equipment, or like just a transfer. What we put the gel and the membrane next to each other, and you run a current, so all the proteins will transfer onto the membrane. Then you take that membrane and then you incubate it with specific antibodies, and then you put a labeled antibody. So this would be the primary and this would be the secondary antibody, and then you add a substrate. If you see a color, that means one of these proteins are reactive, and that's the antigen you're looking for. If you don't see any color, then there is no immunoreactive. This is an example of looking at pork protein, the presence of pork protein. So it will light up If it's present, it will light up because the antibody is specific for that. All right, so it's qualitative for screening a lot of samples. And that's it for immunoassays. Any questions? Yeah. Yes. So once the antigen is bound, then you change the buffer. You change the ion and salt, change the pH, you change the way this antigen reacts with the antibody because they react in a non-covalent action. So when you change the salt concentration and the pH, then you will lose this interaction and they will leave the pump. Very good. Any other questions? Okay, so in the next 25 minutes to get us to 50 minutes, I will cover half of this next topic. And then in class, I'll take 25 minutes, finish everything, and we'll have 25 minutes for games. Okay? All right. All right. So keep on track. If I get super excited, raise your hand and say, hands up. We need to start. Are you gonna time me, Max?
Yeah, I got you. Oh, I'll do it.
All right, time me.
So you said 25 minutes?
25 minutes and then we'll start the review. Okay. So this chapter is kind of so annoying, let me tell you. And I wrote that chapter. Nobody, no author wanted to write this chapter because it had a flower from every garden. It talks about so many different contaminants that could wind up in your food. Contaminants and residues. So there was a lot of work to get this chapter put together, but what I'm going to do is not go over a lot of details, but just give you kind of a high level description of what could wind up in the food that shouldn't be there. So anywhere during the entire value chain, from production on the land to us. So you have, the farming, whether it's plant or animal, you have the distribution, you have the processing, you have the retail shops, and then it comes to us. At any point during this whole value chain, food can get contaminated or can contain residue that is harmful for us. Can you tell me what could be a residue that is harmful for us on the farm? Pesticides. Huh? Pesticides. Pesticides. Thank you. Another one for animals. Huh? Yes, microbial, but I'm not going to talk about microbial here. Another thing that the animals are given, antibiotics. We shouldn't have any meat or milk that has antibiotics. All right, so what else can happen when you're harvesting wheat? What can come out with the grains? Huh? Oh, yeah, it can happen, but another thing that is very common in nature, it's everywhere. Huh? No, well, yeah, but what is something you don't want to eat? Yeah, you can have dirt, but insects, right? Bugs. Wait until I tell you how much bugs you're actually eating. A lot. Okay, so anything that shouldn't be in the food and it winds up in the food are residues of concern or contaminants of concern. That's on the field. But then there is transportation and then there's processing. There could be use of processing aids like nitrites and sulfides. Some people are allergic to sulfide. Nitrites can cause carcinogen formation with heat. You know how you cure heat and sometimes If there is heat involved, you can form microzoates. That's carcinogen. So there are a whole lot of things you can grow fungus or mold. What can come out of that? Huh? Aflatoxin and other types of toxins. So we need to be able to identify, detect, and say, okay, this is the level above which it's not acceptable. Above this level, we shouldn't have pesticides. Above this particular level, we shouldn't have aflatoxin. So for every contaminant or residue, there is a level that we cannot. Sometimes aflatoxin B, for example, should be 0. Antibiotics should be 0. So certain things should not be there. All right. So this is an old one. There is more updated data in the book, but I kept this image because it just illustrates that there are so many hazards that wind up in our food. So in just a short, like 4 years period, 12,064 or 641 hazards were recorded. Yes, microbiological, whoever said bacteria, yes, microbiological is a big component, but mycotoxin is a big component. Chemicals like pesticides and antibiotics are component. There are others, including fraud, adulteration. Can you one example of something that huh? Felony, yes. It is adulteration. Sometimes labeling bisphenol A, for example, comes from plastic. Certain plastic, it leaches into the water or the juice or whatever is that plastic. So now there is bisphenol A3 plastic. So packaging, hygiene, glass, metal, whatever. Okay. So the main categories are pesticides, mycotoxins, antibiotics, and then there are allergens. So these are 4 big categories, and then we have all others with chemical additives like melamine adulteration or presence of sulfide or argon. Okay, this table is a great table to study from, really high level. It just gives you what types of analyses you would do, and if it is quantitative or qualitative. So these are your main, and also GMO is in there. So these are your main residues and contaminants, and these are just high-level methods. Because in an exam, I can tell you, okay, this is the food, what could be there? You want to example what could be a residual concern. And then I will ask you, how would you detect it? You can say HPLC, if you give one line, it's what HPLC is. And how would you detect it? Mass spectrometry or UV detection or fluorescence? Okay. So basically, write this down, okay, remember, this is the food. You want to give an example of a food. If I give you a contaminant, you give an example of the food. I give you fluid, you give me an example of contaminant. How would I detect it? What's the method of analysis? HPLC, immunoassay, to state the method of analysis. And then when I say, what's the detection? Detection is measuring absorption, doing mass spectrometry, looking under the microscope. So this is the tool that you use to detect. So there's the analysis, and then how to detect. In ELISA, what's the detection then? Absorption, huh? Yeah, spectrophotometry. Great. So that's the example. So the immunoassay, you can explain it briefly and then say you need a spectrophotometer. That's the example. So make sure you know that distinction of method of analysis and then detection. This is a great example of a very high level, but I expect you to know what BC does, what BPLC does, cover that already. Okay. So general consideration, most of the residue and present a very small concentration. and they might not be homogeneous in split. So sample preparation and sampling, chapter 5 that nobody likes, very important in this case. Because if we mess up sampling and sample preparation, we will get seriously wrong results that might be harmful. This is the one place where sampling is super important as well as sample preparation. So we need to make sure that sample is homogeneous, is representative, you take multiple regions, and then sample preparation does not cause any alteration of the components. Then we do a series of extractions and purification. Sometimes we really have to do very like stepwise purification to get a concentration that we can measure. But these are present in really small concentrations. Then we determine the method of analysis and the detection based on what we know about this component. So sample preparation depends on the volume sample and its matrix. Does it have high fat? Do we need fat first? What kind of solvents we need to use? So a lot of like homework we need to do before we select the preparation and the analysis. Then we do data analysis and then. Okay, so this breaks it up for you. You know, sample preparation, the homogenization, the extraction, cleanup, meaning, you know, what I really want you to know is like there are series of things, but you know, I'm not going to worry about details here, but I want you to understand that sample preparation is really important, homogenization. Then we need to extract and concentrate, clean up, remove all of the non-essential things and really concentrate. Then analyze different ways of analysis detections. Sometimes if we're using GC, we need to do derivatization. Remember, derivatization in GC makes the compounds thermally stable and volatile if they are not already. All right, so the high level, what I want you to remember, sample preparation, enough homogenization, multiple sampling, extract the component of analysis, concentrate, then determine how you're going to analyze it. So that's the high level that I want you to. Okay, so I'm going to talk briefly about pesticides. So there are so many pesticides, but they have to all be registered. Before a pesticide is allowed to be used on the field, we need to determine the toxicity level, to characterize it and understand how much we can apply that will be safe for the consumer. Once we know this information, that it can be registered, it can be legal to use. And there are ways to analyze. There are the multi-residue. When we know that the farmer used a pesticide that is a cocktail, then we can extract and analyze all of them in one analysis. Sometimes we need to be very, very selective in our preparation to analyze the presence of one. And usually this is done for registration when you really want to isolate one pesticide and study the level that would be harmful for us. So this is mainly for registration purposes. This is for screening different pesticides. Most of them that are used in the field are cocktail of the pesticide that would harm different pests. So we can do quantitation, we can do qualitative. The qualitative is with thin layer chromatography and enzyme inhibition assay. So sometimes we just want to screen. Do we have pesticide? Yes, no. If we have, we take those samples that were positive and we can do quantification. So analysis chromatography, usually GC is used for non-polar pesticides. But now we have a lot of polar pesticides, we use HVLs. So GC is used when it's mostly non-polar, volatile, thermally stable. So we choose columns and detectors. And then we can also do mass spectrometry on them for detection. HPLC is used when your pesticide is polar, thermally labile, not stable, and non-volatile. So you don't want to do derivatization, you just do HPLC. There are different types of columns based on the pesticides used, and then different detector, mostly with pesticide, detector with the MS. The MS is used because it gives us a lot of identification power, but more so tandem MS. So not only one MS, but MS in tandem. Because look at that. Sometimes with one MS, you have really crowded spectra, and your standard, the pesticide confirmation molecule that you're looking for is 231. But there are a lot in your sample that kind of make it hard to distinguish. So we can do an MS, take this 231 and blast it to fragment it more, and then you get a really nice clean spectra compared to the standard. And then you can confirm the presence more clearly than a very crowded single mass spec spectra. So tandem MS is usually to give you more clean, confident spectra compared to a standard. We can do ELISA, and this would be competitive because pesticide molecular weight is small. You cannot sandwich it. They only have one epitope region. Okay, so, and then thin layer chromatography. It's based on presence or absence of a pesticide that inhibits an enzyme. So this pesticide usually they kill pests by inhibiting a crucial enzyme in these bugs. So the way we do it, we separate the samples by thin layer chromatography. We spray with enzyme all of the sheet of the TL, the thin layer. And then we spray with enzyme substrate. So enzyme substrate. And then we see zones of clearance. So wherever you have a pesticide, the enzyme cannot convert the substrate to a product. You inhibited the enzyme. So when you see these zones of clear, meaning the enzyme is inactivated, you have pesticides. All right, microtoxins. So they come from fungus, and there are a lot of different mycotoxins. So aflatoxin is one. Aflatoxin B is the most harmful carcinogen. Should be present at 0 level. Aflatoxin A and other types of aflatoxin, you have a tolerance level. So they're mostly carcinogens. Method of detection, HPLC, thin layer chromatography, absorbance or mass spectrometry even, and then competitive ELISA or immunoaffinity chromatography. So there are so many different methods for determining microbes. Drug residues, antibiotics. So penicillin, amoxicillin, a lot of people have allergy to these antibiotics, so they shouldn't be in food. Also, they shouldn't be in food because they would encourage bacterial resistance. So the bacteria will become resistant to antibiotics, so they won't work anymore. So they are given only if the cow, let's say, is sick. They give therapeutic level, but at the time of, producing the milk or slaughter, there shouldn't be any antibiotics in the system. So, but sometimes the farmers give sub-therapeutic levels to eliminate possible infection and to help with growth. But again, they should be removed by the time of slaughter or their. We don't want these antibiotics because people could be sensitive, bacterial resistance, and potentially, if you have milk with antibiotics, it will interfere with the process. production of yogurt. You don't want those antibiotics to be present, zero level. So there are different ways of determining. Simple ones, if you get a culture of bacteria growing medium, and then you add the sample of the milk, let's say, and measure growth by turbidity. If there are antibiotics, bacteria will not grow, you will have less turbidity. This is a very common one, the diffusion, this diffusion method. So this is an agar, and you have little discs. I don't know if you can see, like little round discs that will put your milk sample in it. And then you do your thing with microbiology to grow the bacteria. And then if there are antibiotics, they will seep out of the disc and you won't see any bacterial growth around that disc. So that indicates you have antibiotics. The most common one is charm test, which is, it works like a competitive ELISA, where you have the antibiotic, the same antibiotic you're suspecting to be present is radio-labeled. And then you put your milk sample and then some bacteria in there. So if you have the antibiotic, it will react with the bacteria. But if you don't have it, the radio-labeled drug will react with the bacteria and then measure radioactivity. So it's compatible. If you have it, you will have less radioactivity. If you don't have it, you will have more. So compatible. The same principle. It's called charm test. And then also you can do quantitative analysis by HPLC or GC with detection by X. Food allergen is another important component because people have allergenicity, the big nine. It means that these are the most allergenic potential components that represent 90% of the allergic cases. Yeah. Huh? Oh, yeah, I'm not. All right, so 90% of allergenic cases are represented by these 9 allergens. There used to be 8, but then sesame was added. And the Allergen Labeling and Consumer Protection Act states that the label should clearly indicate whether or not there are these ingredients or sources in the food, even if it's produced in a plant that might have, like, you know, if you look at the chocolate M&Ms, they say produced in a plant that might have contaminated peanuts. So it is regulated heavily and it has to be labeled. So the way we analyze, we have protein-based method and DNA-based method. Protein is you look at the reaction of an antigen protein allergen with antibodies. We use ELISA, Western blot, or virus sensors in the assay. DNA method, you use PCR just to look to see if the presence of a particular DNA of a source is there. But the problem is sometimes when you isolate an ingredient, you don't have the DNA anymore. And if you put it in, you won't be able to detect DNA. But there are also for proteins, sometimes you're not able to detect the protein in there depending on the extraction method. Let's say here in this figure, they used salt to extract, so they found the antigen. When you don't use salt, you don't find the antigen. So it really, both methods have advantages. All right, how am I doing? You got 2 minutes. All right, so good, the second way to stop here. So other contaminants, the list is huge. We have a table that will be good to study from. But like I said, others could be those that could be present due to processing, sulfide, nitrites, furans, acrylamide. It could be additive or or due to processing, high temperature processing. Melamine is an adulterant, packaging material, and so on. Some are legally banned, some you have to declare on the label, especially for sulfite, for example, sensitivity to sulfite, you have to declare on the label. Some are approved, but they are of concern, so we need to also declare how much there is. And I'm gonna stop here. The rest shouldn't take more than 5 minutes, and then there is also the silk that I wanna talk about that might take 5 minutes. All right, so we can switch gear. Do you want to stretch, take a break? Yeah, take 5.
Going.
Of the